In this module, we will talk about media optimization. But before talking about the media optimization, I would like to tell you about our previous modules. In our previous module, dealing about the media for industrial fermentation, we talk about different important part of the medium. So, just for designing and formulation of the medium, when we decide that the carbon, nitrogen, phosphorus and the minerals, all those are our requirement. But their quantity and the time of the supply, there are so different number of aspects that need to be optimized. Just one case, when we say that the growth of any microorganism in different isolated phase, that require a specific conditions. One is nutritional requirement and other is known as physical requirement. As concerned the nutritional requirement, in which carbon, nitrogen, phosphorus, mineral sources, inducers, precursors, antifoaming agents, all those. But as concerned the physical requirement, aeration, agitation, temperature, etc., these all are the desired requirement just to optimize the growth of any microorganism. So if we say that optimized condition, in order to have a maximum or optimized growth of any organism. So if there is a optimized growth of any organism, because the fermentation is actually the mass culturing of the organism. So that is only achieved when we have all the things in a very optimum level. So optimum conditions is required to set the fermentation process just to make ensure the maximum productivity and minimum cost. So when we talk about just an example that a carbon source. So if we have carbon is the dual role, number one, that is the part of the building block of the cell. On the other hand, that also provides the energy to the cell. So if we have a different choices of carbon sources, suppose glucose, methanol, starch, cellulose, so we have to select which one is the best. So first of all, if we select that which source is the best, then the second thing that need to be optimized, that in what concentration, either 0.5%, 1%, 2%, 3%, 5%, that at what concentration that is required. So if we use a maximum 10%, might be that is excess amount. Then by the use of the excess of that, we can increase the cost of the product. So we need the different condition to be optimized. So media optimization by the classical methods, it's a routine and a very classical methods if we want to optimize any parameters, because these are the different parameters either based upon some nutritional requirement like carbon, nitrogen, etc. On the other hand, on a physical parameters like temperature, like aeration, agitation. If we want to optimize and get a suitable point, so we have to fix all other parameters fixed, and then we have to change one parameter. So this is known as the classical approach. So by this way, if we say that if we want to optimize the fermentation process and their different parameters by classical way, then there is a number of experiments required just to optimize a single process. So if we deal with the optimization process by the classical methods, then just for optimization of a single process, then need a long time and number of experiment. So when we perform the number of experiment, then there is a huge input during the fermentation optimization process. As an example, you can see in this slide, totally when we say that how much will be the number of experiment to be conducted to optimize any process. So you can see here that x powered by n. X here is the number of the levels and n is the number of variables. Just an example, if we say that there are the 3 nutrients, suppose carbon, nitrogen and phosphorus, and there are 2 concentration of each nutrients, then we have to conduct our experiment for 8 trials. Just by putting this formula 2 raised to power 3, and the answer is 8 trial. So just for the 3 nutrients and having their 2 concentrations, we need 8 experiment or 8 trials. In other case, if we have a 6 nutrients and we have to optimize 3 different concentration, you can see that 3 raised to power 6, and total number of experiment required to optimize is 729. It's a huge number. So on an industrial point of view, we have to decrease the number of experiment just to optimize the fermentation conditions. So there are other alternative methods just to decrease the number of experiment, just to avoid the classical approach, because the classical approach is time taken and are laborious, and that involve a huge capital involvement. So in later modules, we will talk about some modern approaches of media optimization just to decrease the number of experiments.